right, so I'm going to go through an overview of Chapter 2 and point out some things that you will want to focus on, and you will have some problems to solve in your practice quiz. So Chapter 2 is about chromosomes and cell division. And there's two types of cell division, one that we call the cell cycle / mitosis, and one that's called meiosis. And those are different types of cell division, and we need to focus on the differences. A complication of studying cell division is the terminology that comes with the cells and the chromosomes. So there's some difficult terminology that you will be working on understanding in this chapter. I do expect you to still know about cell structure and function for prokaryotes and eukaryotes, just basic stuff. And especially there's a big size differential between a prokaryotic cell and a eukaryotic cell. We're going to focus certainly on the genetic components, but I do expect you to still have that basic knowledge. Prokaryotes, you may remember, include domains archaea and eubacteria, or sometimes just called bacteria. And those have no nuclear envelope, and that means they have no nucleus. They do have DNA. It's in a region of the cell called the nucleoid region. Usually the DNA in the nucleoid region is just a single circular chromosome, but they also have in the cell, in the cytosol, plasmids. And it can vary how many plasmids, and there can be more than one kind of plasmid in the same, in one bacterial cell. So those are highly variable, but the species has a particular circular chromosome that's associated with a particular species of bacteria. Eukaryotes, of course, have a nuclear envelope, so the DNA is found in the nucleus, protected by the double membrane, which is called the nuclear envelope. The DNA is wrapped around histone proteins, which helps protect it and also organize it, especially during cell division. Eukaryotes have linear chromosomes. The number of chromosomes depends on the species. So, for example, most human cells have 46 chromosomes per cell, and a mouse has 40 chromosomes per cell. And there are even organisms, there's a type of deer that has 4 chromosomes per cell. There's yeast, one of your model organisms, has 32. Drosophila has 8. So it just varies based on the species. And the trick is that chromosomes don't have to be any particular length. So just because one species has more or less chromosomes per cell doesn't mean they have necessarily more or less DNA per cell, because the size of the chromosomes can be quite variable. And then eukaryotes have extranuclear DNA in the organelles of the mitochondria and the chloroplasts if it's a plant. But in animals, the mitochondria have circular DNA molecules very similar to what you would expect to see in a prokaryote. And then viruses, I'll just mention viruses. Viruses aren't considered alive, but they're important to living organisms. Typically, scientists like to study viruses in parallel with whatever their host organism is. Their genetics tends to be similar to the host organism in some ways. The structure of a virus, it always has some kind of nucleic acid, either DNA or RNA. It can be one chromosome or many. It can be circular or linear. But whatever the nucleic acid is, it's surrounded by some kind of protein, what we usually call capsid, or protein coat. And then some viruses then additionally have an envelope around them, which is a phospholipid bilayer type structure. So a little bit about viruses. We're going to be talking mainly about the cell division in the eukaryotic systems. So I do want you to review the cell cycle. I'm not going to spend a lot of time reviewing this. It's part of your kind of your fundamental prerequisite knowledge. You may remember that there are 4 main phases of the cell cycle: G1, S, G2, and M. And you can see there is what I call an off-ramp for cell division. It's called G0. So a cell can go out of cell division and go into G0 for an indefinite amount of time. And then there are things that can activate the cell to go back into G1 again. So you have these phases. S phase, of course, is when the chromosomes replicate. And M phase is called mitosis. That's when the nucleus is divided carefully. That's called karyokinesis. And then the rest of the cell also then divides, and that's called cytokinesis. By the end of each cell cycle, you have 2 cells that are identical. They're identical to each other, and they're identical to the original cell that started the cell cycle. And so we typically call the cell that begins the cell cycle the parental cell, although I think that's in some ways a little bit of an unfortunate term, because it's just that for this one cell cycle, the parental cell just refers to the cell that begins the cell cycle, and then the 2 product cells that end the cell cycle are called daughter cells. But it's just a way of referring to the beginning cell and the 2 ending cells. So the parental cell and the daughter cells are all genetically identical. The fundamental purpose of all of this kind of cell division is to make more cells of the same type. So it's a cloning process for cells. So in an embryo, the cells are undergoing very rapid cell divisions through the cell cycle, and that can cause growth for the embryo, just more cells of the same type, the same genetic types. And then as the tissues develop, sometimes cells need to be replaced. Death or damage to cells needs to be repaired. So cell division can replace dying cells or damaged tissues. So, but fundamentally, this type of cell division, usually just referred to as mitosis, just generates more and more cells of exactly the same type. So they're identical. Now, there are a couple of terms that we use that you may be familiar with. And in the middle of this slide, you can see these words: diploid, and then I put monoploid, and I put haploid. These words, di means 2, mono means 1, HA, haploid, ha means half. And what these prefixes are attached to is this root word ploidy, ploid. And ploidy is something that a cell has, not a chromosome. A cell has ploidy. And what ploidy is how many sets of chromosomes are in the cell. So a set of chromosomes in humans refers to, collectively, one set of chromosomes would be one copy of each of the different kinds of chromosomes. In humans, that would constitute a group of 23 chromosomes. And you can see in the picture on the screen, the first chromosome is shown in yellow. You have 2 copies of that chromosome. So 1 copy of the yellow chromosome, 1 copy of chromosome 2, which is shown in red, 1 copy of chromosome 3, which is shown in purple. So 1 of each of those collectively makes a set. The other 1 of each of those makes the other set. So when you have 2 sets of chromosomes in a cell, we say the cell is diploid. And if there are 2 copies of every type of chromosome, then we say the chromosomes have homologous pairs. Now, the word monoploid would be 1 set. So 1 set of chromosomes, like I said, would be 1 copy of each of these different sized and different colored, it's artificially colorized, chromosomes in the picture. Haploid means half the number of chromosome sets in the cell compared to what's considered normal. So humans normally have diploid cells. Most of your cells are diploid. Then you have a smaller number of haploid cells. Haploid cells are, in animals, reproductive cells. So in females, that would be eggs, and males, that would be sperm. Haploid cells, and again, I'm using the term to describe the cell. Haploid cells contain one set of chromosomes because the normal cells in the organism contain two sets. So haploid is just half the number of sets compared to what is normal. You only have a few, small group of haploid cells in your body. Because most organisms are diploid, or at least a lot of them are, excluding the plants, it does turn out that the term haploid is usually the same as the term monoploid. But haploid doesn't have to mean monoploid. There are plants that have multiple sets of chromosomes. For example, you might have a plant that has 6 sets of chromosomes per cell. And 6 sets would be 6 copies of each type of chromosome, not just 2 copies of each. And if that organism's cells form gametes, the gametes would be haploid. But if, in this case, haploid would be half of 6 sets, so it would be 3 sets. So in that case, haploid would be the same as triploid. So haploid just means half of whatever the normal number of sets are in the cells from that organism. Now, chromosomes can exist in 2 forms. What I usually call the monad form, or the unreplicated form, is what you see on the left. And on the more towards the right, or more towards the center of the screen, you see a replicated chromosome, which is also called a dyad. So monads are just a double helix of DNA. Dyads consist of 2 double helices that are held together at this location called the centromere. And chromosomes have these locations on them. The centromere is kind of a midsection. Sometimes it's a little bit pinched in there. It's really part of the DNA itself. And the telomeres are the ends. So a monad chromosome has 1 centromere and 2 telomeres. A dyad chromosome has 1 centromere and 4 telomeres. There are proteins which attach to the centromere. These are called the kinetochore proteins. And so typically, I don't get too picky about the terms. If you say kinetochore or you say centromere, fundamentally, those are both referring to something in the same location. Centromere is made up of the DNA of the chromosome itself. The kinetochore are proteins that attach to that centromere DNA. One way that we refer to chromosomes is based on the position of the centromere. So if the chromosome has a centromere right in the middle, and in this image, these are all dyad chromosomes. These are all replicated chromosomes. But if the centromere is in the middle, we refer to that type of chromosome as metacentric. If the centromere is a little towards one side than the other, we call it submetacentric. If the centromere is really far off onto one end than the other, we call it acrocentric. And if the centromere is literally at the telomere, which sounds odd, but it's possible, we call that telocentric. Humans don't have any telocentric chromosomes, but we do have metacentric, submetacentric, and acrocentric chromosomes. So those terms are used to refer to the placement of the centromere, and it helps cytogeneticists to identify which chromosome is which when you're looking at a photograph of chromosomes and trying to make sure that all the normal number of chromosomes are there and identify them. So in the same way